too much of a problem. Okay, so you should already know the basics of chemistry. Microbes, just like us, we're made of all of the same, major elements. Many of them are listed here. We require other elements in small or sometimes called trace amounts. Okay, I'm assuming that you already understand things like the structure of an atom. It has a nucleus, it has protons and neutrons, there's an electron cloud. What is meant by like an element versus a molecule. Different types of chemical bonds, hydrogen, ionic, covalent, and different types of chemical reactions. Okay, so broadly in microbiology, we are obviously concerned about biological molecules, biologically relevant molecules, let's say. Often we talk about inorganic compounds. This typically just means that they lack carbon. They're small, usually structurally simple. What becomes maybe more important or important in different ways are going to be our organic compounds that are typically more structurally complex. So when we're talking about inorganic compounds, the most important one is water. You die without water, microbes die without water, everybody dies without water. Water is absolutely critical. But the reason that water becomes so critical is in part because of some of its really cool chemical properties. And again, you might remember this from chemistry. Water is what we call a polar molecule. So that means that it's a neutral molecule overall. It actually doesn't have a charge. But the way that the molecule is structured, part of it has a charge, part has a negative charge. The other part has a positive charge, kind of a slight positive, slight negative charge. And so if you remember water, H2O, right? Two hydrogens and an oxygen. The oxygen is what we call electronegative. It tends to kind of hoard the electrons. The electrons spend more time with that oxygen. So the pole of the molecule that has oxygen is going to be the pole that's slightly more negative, whereas the pole of the molecule that has the hydrogens, and so you'll notice they don't line up in a chain. It's not hydrogen, oxygen, hydrogen. It's oxygen and then the two hydrogens down here at the bottom. The area where the hydrogens are, they tend to not hold the electrons as often, so that's why you have more of that partial positive charge there. This allows molecules of water to interact with one another. We call it a bond. If I were gonna, pick a fight with chemists, I'd be like, they're not bonded though. It's an interaction, but that's fine. I don't pick a fight with chemists. But it's an interaction. We call it a hydrogen bond. It's the slightly positive part of one molecule interacting with the slightly negative part of the other water molecule. So they're hydrogen bonds. And that makes water really important. So the properties of water that are really vital to all living things are the first two. Water has a high boiling point, 100 degrees, or 212 if you like Fahrenheit better. You are not 212 degrees. So because it is hot, right, before water boils, you never boil, which is nice. You might feel like you're boiling in the summer in Central California, but you technically aren't boiling. And it has a high specific heat. It requires like a lot of energy in order to increase the temperature of water. When you wait for your water to boil, whenever you're cooking or whatever, it takes forever. That makes it an excellent temperature buffer that helps you to maintain temperature even if it is hot. That's part of why, when it is 115, you are not 115. The water is a great temperature buffer, and then of course we have mechanisms to help us cool down, like excreting water and evaporating it, so sweating. Okay. Water has high surface tension. For us, maybe that's not as critically important, But that's kind of the phenomenon. If you're pouring a glass of water and you over pour it, it doesn't immediately pour over the edge. If you're real gentle, it'll form a nice bubble on the top. The water molecules will still stay held together. Eventually, gravity will pull them apart. But that property of surface tension is great for things like water striders. It's what allows them to walk on water. And then this one, we don't say vital to all living things, but it's important to a lot of them. Solid water, ice, is less dense than liquid. Ice floats. And you probably, or maybe you haven't thought about why that's good. Because, well, we definitely don't get that here. But think about a lake in an area that does freeze. And all the kids can go ice skating on the surface of the lake, and that's wonderful. But at the bottom is a thriving ecosystem. If ice sank, lakes would start to freeze from the bottom, and everything would die. Fish don't survive the winter frozen in ice, with a few exceptions. Plants, microorganisms. So the fact that the colder form of water, that ice, will float means that you have warmer water underneath and can support as much life as possible. So it's vital to some living things, but not all. And water, of course, has a key role in a lot of chemical reactions. We can use water also to talk about acids, bases, and salts. So the idea of the way something dissociates in water would make it an acid, a base, or a salt. So if it has a hydrogen ion and it goes into water and dissociates, so hydrogen chloride splits into hydrogen and chloride ions, right? That's an acid. It produces hydrogen ions. Sodium hydroxide splits into sodium and hydroxide. So you have a base if it produces a hydroxide. If it does something else, it's a salt, like sodium chloride. And again, that becomes really important because that gives us our pH scale. And when we talk about pH, the pH scale, we as humans are sort of right there in the neutral zone. Call it neutral, which theoretically means that the concentration of hydrogen ions and hydroxyl ions is roughly equivalent. When you have something that has a lower pH than 7, that means you have more hydrogen ions than you do hydroxyl ions. So it's something that's more acidic. Lower pH, higher acidity. And even in our body, our blood is neutral. Our stomach acid is very much not. So our stomachs are very acidic, and that helps us in a couple of different ways. One, it can help break down food. I would argue more importantly, not a whole lot of pathogenic bacteria can survive acidic pH. So you're eating a bunch of whatever you're eating that has microorganisms on it, most will die in that acidic pH of the stomach. This is one reason that people who are very young or malnourished tend to be at higher risk for some GI infections. Like babies don't have acidic pH in the stomach like an adult does. You ever heard you're never supposed to give an infant honey? Do you know why? Botulism. So the bacteria, the spores, will germinate, and the adult stomach acid can mostly tolerate that, but infants don't have a strong enough stomach pH. But people who are malnourished also have an elevated pH. So you might ask, well, if our stomach pH is 1, why do we even get foodborne illness at all? Stomach acid should kill everything. Well, depending on what you've consumed, right, that could also cause a temporary increase, right? If you've got milk products, food, right? Not every area of your stomach is going to be 1 at that moment, so microbes can sometimes sneak their way through. But it's very, very good for a lot of different things. We also find microbes that have evolved to not only tolerate acidic pH, they thrive in it and they require it. They've altered their membrane chemistry, and we'll talk about that later, to be able to survive in that acidic pH. And remember, when we're looking at this scale and we're going from 7 to 6 to 5 to 4, right? Each one number on the pH scale is a factor of 10. So something with a pH of 6 is 10 times more acidic than a pH of 7. pH of 5 is 100 times more acidic than a pH of 7. We go to the other end, we start increasing the pH, we decrease the acidity, increase the alkalinity, we're going more basic. So something like milk of magnesia that you might take if you have heartburn to help neutralize some of the stomach acid. Bleach, oven cleaner, ammonia. You can use them as household chemicals, but you also don't put like pure bleach on your skin. It'll burn it, just like an acid would burn it. There are organisms that have evolved membrane adaptations that allow them to thrive in these alkaline conditions. There are fewer that we know of compared to acidophiles, but there are some. Most organisms, including the ones that infect us, grow best with a neutral pH. And that makes sense. If an organism requires a pH of 2, it's very unlikely to be able to cause an infection in your blood. If it requires a pH of 7, they're home. You're perfect for them. So what we tend to see is that pathogens, right, have basically a lot of the same physiological requirements that we do. They didn't evolve them to infect us, but they're able to infect us because that's how they evolved. So, that major inorganic molecule is water. Of course, we have acids and bases, and salts that are inorganic. But where we spend the bulk of this chapter is with our organic compounds, because organic compounds are really foundational to building an organism. So an organic compound is going to contain carbon and hydrogen, and often will contain different functional groups. And these functional groups are what give each of the molecules their different function. That's why they're called functional groups. So here we have a really simple set of organic molecules. We have 3 different alcohols: methanol, ethanol, and isopropanol. In theory, people can use 2 of them as like antiseptic agents. You can use ethanol or isopropanol. Rubbing alcohol is isopropanol. They differ in the number of carbons. That's it, number of carbons and hydrogens. You can drink ethanol. You can't drink methanol. Well, you can, but usually only once. So don't recommend it. So even just those subtle changes can really change the property of the molecule. Taking away a carbon takes it from a molecule that some people have a really high tolerance to a molecule that will kill you. Okay, so that becomes really important, talking about those functional groups. So what we're going to talk about in this chapter are the biological macromolecules, polymers of small organic molecules. So, like methanol is a monomer. Even glucose is a monomer. It's a single subunit that has its own chemical properties. But what we talk about is let's join them together. Let's join a chain of glucose or a chain of other sugars. And we get our carbohydrates. We have an amino acid. We join them all together. We get a protein. We have adenine or thymine or guanine or cytosine. We chain them all together and we get DNA. There are lots of different lipid ones, so I can't think of a real quick, fast example, but you have lipids too. So those are our 4 biological macromolecules: carbohydrates, lipids, proteins, and nucleic acids. So we take an individual subunit, a monomer, and we join them together. It can be the same monomer, it can be different monomers. And when we join them together, I told you water will be very important for all of these reactions. When we join them together, we produce a water at the end. These reactions are either called dehydration synthesis, because you have dehydrated the original molecule, you pulled a water out, or condensation reactions, right? You produce water, it's condensation. You can call them whichever one you want, I don't care. I give equal consideration to both, so whichever one you remember and whichever one you like the best, that's fine. These are covalent bonds. So they require energy to build these molecules. Okay, so our first class of biological macromolecules that we'll talk about are the carbohydrates. So carbohydrates generally have 2 really broad functions in living systems. They can be used as energy. We typically call those sugars. They can be used for structure. We call those starches. But we can also use some as components of other macromolecules. For example, when we talk about nucleic acids, we'll see that we require a sugar to make a nucleotide. So you can use one biological macromolecule as the foundation for another. Carbohydrates will have a consistent chemical formula, CH2O, repeating however many times. A lot of them are what we call isomers, meaning they have the same chemical formula but different structures. So how's the molecule put together? So you have things like D or L glucose. Here they show you glucose and fructose. They have the exact same chemical formula, C6H12O6, but the molecules put together a little bit differently. So when we're talking about a monomer of a carbohydrate or monomer of a sugar, we call those monosaccharides. These are simple sugars. They'll have anywhere from 3 to 7 carbon atoms. We do spend a lot of time talking about a very particular 6-carbon sugar, glucose, but here I show you fructose as well. We also spend a lot of time talking about a 5-carbon sugar, deoxyribose, which is the sugar component of DNA, and ribose, which is the sugar component of RNA. But of course, those are not the only important sugars. We can join sugars together, 2 monosaccharides, through dehydration synthesis or a condensation reaction. So here they're showing the joining of glucose and fructose. When you put those two monomers together, you get table sugar, sucrose. In the body, one of the first things that can happen is we want to process this, so we need to break it down to its monomers. We need to reverse that synthesis. We use water to break the bond, so you can follow the reaction from left to right or right to left, however you want. When we use water to break a bond, we call it hydrolysis. Use water to break a bond. Lactose is a very common disaccharide. Glucose and galactose are the two monosaccharides that make up lactose. And then, of course, we have polysaccharides, long chains of monosaccharides. And they can all be used for different things. They'll have structural differences depending on how the different monomers are joined together. So like, are the linkages like this, or are they more like this, or does it look a little bit more like that? Okay. There are, again, important polymers of glucose. Starch is an example. Glycogen, we use glycogen as storage. We can store sugars for energy in glycogen reserves. So do bacteria. They can sometimes form these big inclusions that are just sugar for them to use later. Chitin, the exoskeleton of insects or the cell wall of fungi. Agar, that you use to solidify your plates. They're all carbohydrates. Questions about carbohydrates?
Wait, so chitin is also made out of like the exoskeleton of insects and cell wall? How does that work?
It just works.
It just is?
It just is. Just chitin. Some fungi have, they use mannan or glycan, but chitin is also really common, and chitin is the exoskeleton as well. Other questions about carbohydrates? So again, what's going to be important for this section is, can you tell me what the biological macromolecules are, what their function is in us and in microbes, and what are the basic units? I'm not saying you have to remember that glucose plus fructose equals sucrose, but you should remember monosaccharides, disaccharides, polysaccharides, dehydration synthesis, broken up through hydrolysis, things like that. What they're used for in the body, including the microbial body. Okay, then we have lipids. Okay, carbs get a bad rap because nobody wants to eat carbs. Low carb, keto, whatever. If you don't have carbs, you're going to die. So you have to eat some. Lipids are the same way. Fats, I don't want to be fat. Like, don't eat fat. You have to eat fat. You have to. You have to have it. Microbes have to have it. You have a whole thing that surrounds each one of your cells called a phospholipid bilayer. Good luck without that. Lipids are absolutely essential for cells. They are the primary components of the cell membrane. They are also really useful for energy storage. That's why, in multicellular individuals that have an excess consumption of calories, that energy is stored as lipids. They're great energy storage molecules. They're so good, that's also why when people are in a calorie deficit, you lose the lipids, you lose the fat, because the body is like, oh crap, I need energy, and starts breaking down those best energy storage molecules. So lipids can have structural function as like the cell membrane. They have energy storage. They also work as things like hormones. Many hormones are lipid derived. They also do consist of carbon, hydrogen, and oxygen. They will often also have different functional groups depending on what kind of lipid it is. We can break them into two kind of groups, simple and compound lipids. So they're non-polar, they're insoluble in water, which is why, like, if you mix oil and vinegar to make a salad dressing, you really have to shake it up, because otherwise it would look like that, and that's probably not what you want to have on your salad, because otherwise that's what it'll do unless you mechanically force it to integrate. So this is an example of a simple lipid, a fat. This is what we call a fat, or a triglyceride. So it's called a triglyceride because we have 1, 2, 3 glycerols at the top. So it contains a glycerol, right? It has 3 chains. So it's 1 glycerol with the 3 chains, the 3 carbons, 1, 2, 3, each gets a fatty acid tail. So glycerol plus a fatty acid is a triglyceride. And these again are what we typically call our fats. So when people are talking about the fats that you're eating, that's what they're talking about. The chains are formed by dehydration synthesis. Nothing fancy. But the reason that these are so good for energy storage is, as I'm sure you remember from chemistry or your other biology classes, when you break a bond, it releases energy. Look how many bonds you get to break. You get a lot of energy from breaking up fatty acids. You'll hear people talk about things like saturated versus unsaturated fatty acids. We can talk about it in the context of like human nutrition, but we can also talk about how, and we'll talk about that on the next slide as well, how that can affect microbes as well. So when someone is talking about a saturated fatty acid, what they mean is that every single carbon of the fatty acid tail has as many hydrogens as possible. It is completely saturated with hydrogen. There are no carbon double bonds. An unsaturated fatty acid, this one right here, has a carbon double bond. So in theory, there aren't as many hydrogens as could be supported by the number of carbons. If these carbons were only single bonded to one another, right, you could put another hydrogen on each of those carbons. So an unsaturated fatty acid does not have as many hydrogens as it could. Okay, so you'll notice here, they also have an arrow and they specify cis. So when we talk about unsaturated fatty acids, we can talk about cis fats and trans fats. And you all might be too young to remember the trans fat panic. There was a trans fat panic. And it just has to do, well, you know from chemistry, cis and trans are, right? Do we have the same, right? The functional groups come off the same side of the carbon double bond. That's cis. Or do they come off opposite sides? That's trans. So that's like chemically what's happening. Biologically what's happening is saturated fats are bad, quotes, right? Because they can pack very tightly together. You can pack a lot of straight lines together. Cis unsaturated fats are nice because they start like kicking, right? They kick out this little bottom part. So you can't pack as tightly together. Trans fats are bad because they form a little zigzag, which is easier to pack tightly together. So the idea is that of the fats, unsaturated, polyunsaturated fats, especially with cis, are supposed to be the best because they just don't pack as tightly together. And I mean the best in terms of like they're less likely to clog your arteries, so in terms of cardiovascular health. But again, on the next slide, we'll talk about why microorganisms might skew one kind versus the other. It's not really good or bad. It's just, it is what it is. Okay, so saturated fats and trans fats can pack really tightly together. Cis unsaturated or polyunsaturated can't pack as tightly together. And again, in people, we think if they pack less tightly together, that's good. I'm less likely to get clogged arteries. But in microorganisms, maybe we think a little differently. So the complex lipid that makes up the phospholipid bilayer looks a lot like a triglyceride, with a little bit of a difference. So remember, the triglyceride has the glycerol and then the 1, 2, 3 fatty acid tails. Each carbon of the glycerol is attached to a fatty acid tail of different lengths, saturated, unsaturated, whatever. In the phospholipid, you've still got the glycerol, so you have 1, 2, 3 carbons. 1 and 2 have a fatty acid tail, but 3 has a phosphate group. And then maybe something else, like an organic group on top of that. So that's your phospholipid, and that's the difference between the phospholipid and the triglyceride. So you'll notice they still do illustrate, the 2 fatty acid tails. They show you one that's saturated, one that's unsaturated. But now they put it together in a biological membrane. So what does that look like, since those fatty acids could be saturated or unsaturated, just like the triglycerides? So what does that look like in a biological system? Well, if the majority of your phospholipids have saturated fatty acid tails, you get a pretty closely packed membrane. Whether that's good or bad, we'll talk about in just a minute. Unsaturated fatty acids, you see all the whatever's going on there in the tails, that makes it less packed together. It's more loosely organized or loosely packed. So why is that important? Why do you care in a microbiology class? Well, again, we exist at our temperature, and pathogens that infect us exist at our temperature. Many microbes do not exist at our temperature. Microbes that exist at colder temperatures, well, what happens when fats get cool? They solidify. So you're a microbe that lives at 4 degrees or colder. Your membrane is basically fat, and now it's solid. What's the problem if your membrane is solid?
Nothing will pass through.
Nothing will pass through. Right? Your membrane is a very dynamic system. There are proteins constantly moving around the surface. You're trying to do transport of whatever you can. Sometimes things can just pass through. Sometimes you need to bring things in right through protein transporters. And now it's goo. And you can't move anything through. So microbes that live in colder temperatures have evolved membranes that contain a lot more unsaturated fatty acids. You still retain that fluidity as the temperature decreases. So now we can see why microbes that are cold adapted might have a higher percentage of unsaturated fatty acids. So what about what happens when you heat fat up? It becomes a liquid, right? It starts to move around a lot. So if you're a microbe that's heat adapted, you want more saturated fatty acids, right? Same thing like when you boil water. Eventually, things go from solid to liquid to gas. You start to get too much movement and too much disorganization in the membrane. So the more saturated fatty acids you have, it's not that you retain fluidity, it's that you're able to hold yourself together. At least the membrane part. You're able to hold your membrane together. So of course, there are a ton of biological implications in humans when we talk about the difference between saturated and unsaturated fatty acids. But for microorganisms, this is absolutely critical. How have microbes evolved, or what evolutionary mechanisms do they have that have allowed them to thrive in ice or in a hot spring? And adjusting the percentage of saturated versus unsaturated fatty acids is one, but not all, one such mechanism. So it's really important that we understand that about lipids. They're not just, fats are bad. They have a lot of important biological relevance. And then, of course, there are other types of lipids. I mentioned, for example, hormones. Some hormones are lipid derivatives. Things like the steroid hormones, those are lipid derivatives. Cholesterol is also a lipid. It also can be embedded in the membrane and play a role in membrane fluidity. So again, like from the human perspective, we always talk about, oh, cholesterol bad, like your arteries are gonna be all clogged. Yeah. But from a biological perspective, again, it increases fluidity at lower temperatures. So not only do cold adapted microorganisms have more unsaturated fatty acids, they often have more sterols, like cholesterol, in their membrane to retain that fluidity. And of course, on the other end, you would expect that most heat adapted microorganisms would lack as much cholesterol. And again, sterols in the membrane are very, very important. Is it always cholesterol? No, fungi use a derivative called ergosterol. But something like ergosterol, like having a sterol, that lipid derivative as part of the membrane, is so important that actually some of the best antifungal drugs we have inhibit ergosterol biosynthesis. They can't make their lipid sterol, and so they die. So it can be very important to have those sterols in the membrane. So these are all different kinds of lipids. In some ways, I guess I would argue that lipids might be the most diverse of the biological macromolecules, because we do have like the triglycerides, we have the phospholipids, we have the steroids. Whereas with something like a carbohydrate, you have a monosaccharide, disaccharide, polysaccharide. How you link them together, you get a lot of different carbohydrates, but overall, right, the single unit is the same. So lipids are a pretty diverse category of biological macromolecules, but hopefully you appreciate their importance a little bit. Okay, questions about lipids? Okay, well, everybody loves protein. Proteins are the next biological macromolecules. I guess if we're building in complexity, the base unit of a protein will have carbon, hydrogen, oxygen, and nitrogen. And this is not what we saw with carbohydrates or lipids. And sometimes they have sulfur. Yeah, sometimes they have selenium, but that's okay. What do proteins do? Well, basically everything. They can have roles as enzymes, so they can catalyze chemical reactions. We mentioned that they can be really important as being part of the membrane to bring things in or take things out of the cell, so transport. In human medicine, a lot of bacteria produce toxins that are proteins. And of course, they're a major component of cell structures, including the bacterial flagella. It's all protein. Part of the cell wall, peptides. That's why it's peptidoglycan. So proteins, very, very important. The monomer of a protein is an amino acid. And it's called that because it has an amino group, right? That's the nitrogen part. Amino acids have the same basic structure. And of course, like not for this class, but when you take a class like biochemistry, you have to memorize all of them. So something to look forward to. But just kind of understanding that you have an amino group and a carboxyl group. Those are the two, I don't want to say main functional groups, but kind of. They're the main functional groups of an amino acid. You can't make polymers, you can't make a protein without those. Amino group and a carboxyl group. So you've got the two carbons, and you've got that nitrogen. You'll see attached to this carbon right here in the middle that is neither the amino group nor the carboxyl group, you have an R group or a side group. This is what gives each amino acid its different properties. So they all have the same backbone, but we just swap out what's in the middle. And sometimes what's in the middle is really simple. It's a hydrogen. Yay. When they ask you that one's pretty easy. And sometimes it's less simple. You have tyrosine. So, commonly in microorganisms and in us, there are 20 side chains. 20, so it gives us 20 amino acids. Theoretically, or in practice, I guess we say 20 is most common. Eukaryotes are known to have 21, and prokaryotes actually have 22, but they're far more rare. So again, I told you they can be as simple as you added an extra hydrogen, here's glycine. That's like the simplest amino acid. And I'm not going to ask you to remember the structure. I promise you will have to memorize them for biochemistry. It was really annoying. I memorized all of them, and then my professor, one question, draw glycine. I'm like, oh, you've got to be freaking kidding me. Studied all of these. Do I remember them now? Nope. I remember being angry and drawing glycine. But those 20 different side chains, again, are what give each amino acid its own properties. And the way that you start to link those together into proteins dictates the three-dimensional shape and the function of that protein. They can exist in stereoisomers. They have chirality because they have that carbon that can have 4 different things attached to it. So you can have L or D stereoisomers. L is most common, except for when we're talking about peptidoglycan, where they use the D form. So it's like that handedness. Up at the top, you can start to classify the amino acid side chains. You have nonpolar, you can have polar, and then where am I charged? Where did my charge go? Aspartic acid, glutamic acid, right? You have some that are charged as well. And again, proteins end up being very sensitive because structure dictates function. And as we talk about like control of microorganisms, a lot of the mechanisms we use to control microbial growth involve affecting their proteins, especially the shape. If the protein loses its shape, it loses its function. And that eventually kills the cell. So that's why we often use things that denature, change the shape of a protein, as an antimicrobial mechanism. When we are joining 2 amino acids together, we do so through dehydration synthesis or a condensation reaction. And it forms a very specific type of covalent bond that we call a peptide bond. So in order to form the dipeptide, right, you take the carboxy terminus of amino acid number 1 and the amino terminus of amino acid 2, right, and that's what gets joined together. So they'll call them the amino terminus or the N terminus, or the carboxy terminus or C terminus. The front and the back. The front is the N terminus, the back is the C terminus. And they always have to be joined that way. So like you're not gonna have one come in up here at the front and join in front of glycine. You can add to the back of alanine, you can add to the back of that one, the back of that one, back of that one, back of that one, but you don't add the other way. Just the way the enzymes work.
You said N is always to the back.
N is the front.
N is the front and then
C is the back.
Okay.
Yeah. I know they're in not alphabetical order. It would be helpful if they were in alphabetical order, but no, unfortunately. But it is. You see it's a condensation, right, reaction, a dehydration synthesis. We pull the water, but this specific bond between each amino acid is called a peptide bond. And so like I was saying, this idea of all of these different side chains is how we go from a chain of amino acids to an actual functional protein. And oftentimes it has to deal with how do these various side chains react with one another. Are they not going to interact? Will one of the positively charged amino acids form an ionic bond with one of the negatively charged amino acids? Will the sulfur group of cysteine or methionine form a covalent bond with the sulfur group of a different cysteine or methionine? The hydrogen bonds between the different side chains? All of that dictates protein structure. Many proteins are capable of folding spontaneously. They just fold. But others require the help of another protein called a chaperone. So chaperones can assist in protein folding. Chaperones can also assist if something is going on in the cell that is causing proteins to misfold. So there's a whole category of proteins in bacteria called heat shock proteins. You imagine what turns them on. Heat. It gets too hot, we start making these proteins because they are chaperones that prevent the heat from denaturing the protein. So chaperones can either help fold the protein or help it maintain being folded under conditions of stress. So when we talk about protein structure, we talk about the, 3-4-ish levels of protein organization. Just straight up off the ribosome, you have a chain of amino acids. That's the primary structure of the protein. It's really not even structure at all. It's just the sequence of amino acids. Now, eventually, you can start to get some hydrogen bonds that will form maybe between side chains, between the carboxy terminus of one, like the little carboxyl group hanging off, got that little oxygen there, and a hydrogen sticking out somewhere else. So you have these hydrogen bonds that are forming, and you'll get what's called the secondary structure. We have two really common motifs of secondary structure. The alpha helix, and to me that always looks like the little phone cord, right? A little alpha helix, or a beta pleated sheet. Again, these have biological importance, not only because, they help us fold proteins, but alpha helices, so regions of a protein that have an alpha helix, are a little bit more flexible. So like when we have an enzymatic reaction, it's not just this really static thing. Enzymes will move in order to grab and process the substrate. And again, when you're cold, things don't move as well. So we find that most microorganisms that live in cold environments have more alpha helices in their proteins than beta sheets. It gives a little bit more flexibility to the protein. We see beta sheets when we need less flexibility. When you start putting all that together, and now you have maybe like, again, those ionic bonds I talked about, those disulfide bridges, ionic and covalent bonds, these interactions between the side chains, you get the three-dimensional structure or the tertiary structure. Many of our proteins, that's it, that's where we go. You have a single polypeptide that is made, it is folded into its three-dimensional structure, and that is your final product. But sometimes we have a protein that's made of multiple subunits. So there are independent polypeptides. It could be identical subunits, it could be different subunits that all have to come together to make the functional thing. So hemoglobin has 4 subunits. So each subunit does primary, secondary, tertiary, and then the subunits come together to make functional hemoglobin. We call that quaternary structure or 4th level structure. Dinitrogenase, that's the enzyme that bacteria use to fix nitrogen. They pull nitrogen out of the atmosphere and turn it into biologically available nitrogen. It has 2 non-identical subunits. So if to get the final functional protein, you need multiple polypeptides to come together, you would have a quaternary structure. Less common than just the tertiary structure. So you can imagine when you start to think about, well, there's 20 amino acids, 22 if you're being pedantic about it, and you can make proteins of varying lengths, you already come into this idea that there are like thousands of potential proteins in a cell. But then you can modify those proteins, which gives them different functions. So they can be modified through the addition of other chemicals. You can add carbohydrates to them, you can add nucleotides to them, you can add lipids to them, you can add phosphate to them. They can be methylated or acetylated. They can be ubiquitinated. All of these modifications can give different functions or serve as different signals for what that protein should be doing or what should be done to that protein. So then it turns out that there are like millions of proteins when you consider all of those modifications. Right. So they say in humans that we have approximately 20 to 25,000 genes and between 400,000 and a million different proteins when you start to consider all these modifications. And then things like alternative splicing and stuff. So for a long time, people thought that proteins must be the unit of heredity because they're so complex. And obviously we are so complex. We must be made of a very complex set of instructions. But again, like I said, this idea that structure and function are super tightly linked is super, super important. You lose structure, you lose function. And we use this, we take advantage of this to control microorganisms. Well, what do you do in the lab when you're after you finish streaking on a plate? You have your flame and you put your loop in the flame, right? Flames. What do you do if you don't want, a hamburger to give you diarrhea? Hopefully you cook it. You may not, you live on the edge, that's cool. Heat works really well. And what does your stomach do? Low pH, very acidic. What does bleach do? High pH. All of those things in different ways can act to denature, change the shape of the proteins. Alcohols, detergents, right? First thing you do when you get into the lab, and if you don't do this, you better do this or they're gonna yell at you. You spray the green stuff, right? Clean your lab bench. The phenol, right? The rocol. It's a phenol derivative. It denatures proteins. So all of those things can act to denature proteins, which eventually will kill the cell. Okay. And it's very uncommon that that's reversible. You can't unboil the egg. You boil the egg, right? You get it to whatever temperature for however long. And you get your nice hard or soft boiled egg, I don't care, whatever. Put it back in the refrigerator, it doesn't get runny again. You don't unboil the egg. And almost always you don't renature the protein. If you don't want to die, you try as quickly as possible to make more and hope that there's not still that same pressure that caused your proteins to denature. Chemically, we can renature proteins. It's difficult. I don't like doing it. I've done it before, but you can. Milk is an example where sometimes there can be enzymes that will cause it to, kind of go back to normal. Where if you start to heat up milk, it gets kind of weird, and I don't know, and then you can cool it down and it kind of loses that skimmy weirdness sometimes. Right? But we don't typically see that in nature. You don't unboil the egg. In a lab, we can. So that's why targeting proteins works so well when you're trying to get rid of microorganisms, because they can't renature. They don't have like this complex set of chemical reactions that we could do in a lab to renature all their proteins. And they can't make new proteins fast enough. So as long as you apply the right temperature for the right amount of time, they're dead. The right pH, they're dead. The right concentration of a chemical, they're dead. So it works really well to target their proteins. Okay. Protein questions. Last biological macromolecule, not least, DNA. More complex, I guess the monomers might be here. Because the monomer of a nucleic acid or a nucleotide has 3 major components. It has a phosphate group, a 5-carbon sugar, and a nitrogenous base. So if you have those 3 things, you have a nucleotide. 1 or more phosphate groups, because you could have like 3, like ATP. So 1 or more phosphate groups, a sugar, a 5-carbon sugar, and a nitrogenous base. The 5 most common nucleotides are shown here: guanine, adenine, thymine, cytosine, and uracil. Uracil is only found in RNA, thymine is only found in DNA. Okay, so DNA is, the blueprint for life, our instruction manual, blah, blah, blah. The sugar is deoxyribose. So it's a 5-carbon sugar that is called deoxyribose. In our bodies, DNA exists as a double helix. So you have 2 strands of DNA that are complementary, right, but anti-parallel. They run in opposite directions. A binds, and this is the same for microbes too. A binds with T, G binds with C, and the order of the bases is how we make our proteins. That's the genetic blueprint. All cellular life uses DNA as the genetic material. Viruses are weird and they can do other things. All cellular life uses DNA as the genetic material. But we, cellular organisms, do use RNA for different things. We commonly think of RNA as that intermediate between DNA and proteins when we're doing transcription and translation. RNA can have structural roles. It is a big component of the ribosome, ribosomal RNA. It can have some enzymatic function, ribozymes. In the cellular organisms, it typically is found only as single-stranded. In fact, oftentimes our cells recognize double-stranded RNA as a pathogen signal and will destroy it, because viruses can have RNA-based genomes. RNA or single or double-stranded. The base pairing rules are very similar. In this case, because we don't use thymine, we use uracil. Adenine will bind with uracil. Cytosine will bond with guanine. We'll talk a lot more about how that works and kind of the chemistry of that when we talk about DNA replication. But of course, there are the 3 major types of RNA. mRNA, which serves as that intermediate between DNA and proteins for protein synthesis. tRNA, transfer RNA, which brings amino acids to the ribosome as we're making our polypeptide. And then rRNA, which has that structural role. So what is the difference between ribose and deoxyribose? Right here, you're looking at ribose. So it has 5 carbons, 1, 2, 3, 4, 5. Deoxyribose basically looks exactly the same. The only difference is at carbon number 2. Ribose has a hydroxyl group, Deoxyribose lacks the oxygen. It has only a hydrogen there. That's the difference between ribose and deoxyribose. And so we classically talk about nucleotides and how important they are for, our blueprint for life, DNA, RNA, woo. But ATP is also a nucleotide, which, I didn't really appreciate until I took a class like this that really was like, hey, they're nucleotides. I'm like, oh yeah, that actually makes sense. You've got a nitrogenous base, you've got ribose, you've got phosphates. Okay, so ATP is ribose, adenine, and the 3 phosphate groups, and then we can break, right, the bonds of the phosphate groups to release energy. So they store chemical energy, and they are broken by hydrolysis, which releases energy that we can then harness to do things like make more biological macromolecules. So, classically you would be looking at ATP, you add in some water, and you now have ADP, an inorganic phosphate, and the release of energy. You could do it one more time, make AMP, release some energy, and another inorganic phosphate. So we use nucleotides for energy storage as well. Okay. Questions about biological macromolecules, because this is the end of the chapter. Cool. Hopefully it was a review for most of you. Maybe you applied some of the concepts in a little bit of a different way than you did in another class, and that's great. So yeah, I will see you all on Thursday, where we will start microscopy, which is chapter 3. The slides are already available for you, so go ahead and make sure you download those before you come to class on Thursday.
Are you gonna post the homework today?
Yes.